Hey guys, welcome to episode 3 of my biology lessons, and today we're talking about enzymes. So the role of enzymes is to speed up the rate of chemical reactions by lowering the activation energy. So remember our previous lesson, which was bioenergetics? So look at this hump over here. This hump over here is called activation energy, and sometimes we need a lot of this activation energy in order to get a chemical reaction started. So an enzyme can not only speed up the chemical reaction, but it also lowers that activation energy. Okay, so that's the purpose of enzymes. Now we're gonna get into the structure of it. So this itself is the enzyme. You see that little blob looking shape? It doesn't always look like that, but this is an example. So this entire blob is an enzyme. This purple part, the purple pen part over here, that is the active site, which is where the reaction occurs. And then the reaction is when the substrate, which is the new product, it binds to the enzyme. So when it's like a puzzle piece, when you fit the substrate puzzle into the enzyme piece, the spot where you fit it in is the active site, and then the pieces are enzyme and substrate. And then the substrate is the reactant. So enzymes are substrate specific, which means they can only bind to certain substrates. Enzymes and their specific substrate, they must fit together. So basically, shape matters. And there has to be a certain type of enzyme for the substrate to fit into it. And then we can go into the two hypotheses. So we have lock and key, which is when the active site is it perfectly fits the substrate. So over here, pretend this shape it fits exactly into that. That'd be lock and key. It just fits like a puzzle. And then induced fit is when at first they don't fit perfectly, but hydrogen bonds adjust the shape of the enzyme, and then so the enzyme can fit around the substrate. So basically lock and key is a perfect fit, and induced fit is when it doesn't fit at first, but the enzyme changes in order for it to fit. And then we can look at how enzymes work. So say we have an enzyme here, and this is a substrate here. And you see how this shape basically is a perfect fit? That'd be an example of lock and key, by the way. So in the reaction, the substance just goes into the enzyme, and then over here is this line over here, that's the activation site or active site. And then the process happens, and then once you're done with the chemical reactions, you get the products, and then they just fly off. And then also these enzymes can be reused. Okay, so now we're going to talk about biochemical pathways, which is an ordered series of enzyme catalyzed chemical reactions. So basically, it's a step-by-step process. You have a substrate here, and then it goes in here and it releases the products, and then this product goes into another enzyme and it gets transformed again, and it's basically a step-by-step process. There's multiple enzymes involved. If you guys have any questions on that, because I didn't really explain it that well, just ask me. And then denaturation, we already learned about this, but it's permanently changing the shape of the enzyme. So if you have irregular temperature or pH, then it could denature an enzyme, and then the substrate won't be able to bind to the active site because the enzyme has changed shape and it's denatured. So in order to regulate an enzyme, you need to have the perfect temperature, perfect pH, and you have to have special signal molecules, which is called allosteric enzymes. And then outside the normal range of all of this stuff makes an enzyme denatured. Okay, now we're going to talk about the allosteric site. So the allosteric site receives signal molecules that change the shape of the active site. And then this signal can turn the enzyme on and off. So think of like a on and off switch. So like we have the enzyme over here, right? This is the active site, which is where the substrate goes. And then this over here, like this is where the allosteric activator goes. This is the regulatory site. And then you have an allosteric activator. And then if you put it in, then like you can turn this enzyme on. So like right now the enzyme is off and it doesn't work. But if you put in the allosteric activator, then it starts working. And then you can also inhibit it, which means you turn off the enzyme. So right now the enzyme is currently working, but if you put in an inhibitor in the allosteric site, then the enzyme shuts down and this active site will like change shape and like it won't be able to fit the substrate. Okay, and then now we're going to talk about feedback inhibition. So it's like a controlled pathway of enzymes, and like basically it's here. I have this I have this drawing over here, and I'll try to explain it. So we have the substrate here, and we have the enzyme here. So we can put this substrate here, and then after it goes through the chemical reaction, it turns into this. So like you put this in the enzyme, the chemical reaction happens, and then the triangle turns into a circle. And then next we have the circle here, and then we can put the circle into a new enzyme. And then after it goes through a chemical process in the enzyme, it changes into the rectangle. And then we do the same thing here. We put the rectangle in, it changes, and we get a heart. And then so this is the last enzyme in this set. And also this sequence of enzymes is the biochemical pathway. So like this is an entire pathway. But in feedback inhibition, like this heart can also act as like the inhibitor. So basically if you put the heart, the final product of this pathway into the first one, like, and you put it in the allosteric site, then it'll stop the first one, right? It'll stop this first enzyme from working. And then if it doesn't produce, if it stops working, then it can't produce the circle. And if it can't produce the circle, then the rest of these also don't produce. So basically, it stops this entire pathway. And that is feedback inhibition. All right, so that concludes my lecture. If you guys have any questions, just text me. Thank you.